Okay, so I will go through so how we are using this high throughput proteomics workflow using the Covaris adaptive AFA technology to increase the sample throughput and help our project's needs. So basically, so I'm actually Uthpala Seneviratne from AstraZeneca. So I'm an associate principal scientist in the chemical biology and proteomics group. So our group constantly evaluate automation technologies, new automation technologies, and select appropriate technologies, including the mass spectrometry, to fulfill the project demands. And considering, most importantly, considering cost, because when you analyze a lot of samples, you need to make sure your cost is acceptable and increase the internal capabilities to impact the projects. So mainly recently, our group is tasked to gave us a challenge to help with this DMTA cycle. So DMTA cycle is in the drug discovery in the very early phase, so which is design, make, test, analysis cycle. So which help the early compounds to make suitable, so you can apply to make tool compounds to make in vivo studies, so that we can measure safety profile for these compounds. So it's an iterative process. So when the DMT assays are high throughput, and from that assays we determine IC50, so that means a dose response curves, EC50, so which is the potency, DC50, degradation, or the Dmax, how much degradation is occurred for all these compounds. So as you can see, it is a high throughput process. So when you want to do the high throughput process, our immediate goal is going from 96 well plates to 384 well plates. And I put a quick question. So these DMT assays are typically 10-point dose response curves. And a question for you is how many 384 well plates and how many compounds in a typical drug discovery process we use in a DMT biweekly assay. So these biweekly assays are usually biochemical, or sometimes it's IF immunofluorescence assays that the teams are processing. So I put a couple of answers, a couple of examples, and I can give like 5-10 seconds just to guess what could be the answer, how many plates or how many compounds that usually we are doing in the drug discovery phase. So X is how many plates, how many 384 plates, and Y is the how many compounds. Any for number 1, number 2, number 3, yeah, number 4. Okay. Actually, the correct answer is number 2. So that is the throughput, general throughput. We need 3 plates, 384 plates, 90 compounds per every 2 weeks. It's an iterative process. And chemists use this, the data from these assays to go to the next iterative process. So it's a cycle. So that's why we call it a DMTA cycle. So as you can see, we cannot rely on 96 well plate. So we have to go for 384 well plates if we want to do DMTA assays in the drug discovery scale. So going from 96 well plate to 384 well plate, so that means we can increase the compounds. We can usually, in the mass spec experiments, you can put 28 compounds per 384 well plate. So each compound has 10-point doses. And from the mass spec wise, the added advantage for the DMTA assay compared to the classical assays are, from the mass spec, we can measure multiple targets. It's a multiple target assay, not only one target per assay. And when we develop the method, we also are curious how quality our samples are. So we do deep global proteomics just to check our targets are present. It's a quality control test. Targets are present with the low input material, because we are now in the 384 well plate. And most important factor is the cost. So we need to reduce the cost from going from 96 to 384. And so at the moment, we are less below $3 per well. So you can imagine 384 is how much cost. in this scale. And I will show you, we now have this workflow we can complete in 1 day. So that means we can run 384 in 1 day. So that means we can use it for automation. So it's automation ready. So when you put it to a Hamilton like a liquid handler, so it can process more flow and more plates. So we can now, we predict we can do 4 384s within 24 hours. So that means 4 times 400 samples, like 1200 samples roughly within a day. And most importantly, so I will show you another added benefit is we reduce the consumption of plastics. So it also helps the Mother Earth, and also the cost factor helping us with the reduction of plastics. Okay, so going to the method, so we use this Covaris LE220R system for this process, and the 384 well plates are from Covaris. So here, another added advantage is, so the process we did for these plates are, we first added the compounds to the plate. So we use ECO system, it is acoustic compound transfer system. We use that ECO system to transfer compounds to the 384 well plate, then put the cells, incubate the cells, and wash the cells out, centrifugation, and add the lysis buffer, heat for 95 degrees. It's increased for the increase of the lysis, and then use this Covaris sonication 75 scan method, which lasts for 24 minutes. So with this method, then the next step is digestion. So we again used Covaris instrument to do the digestion for 2 hours. And so that means we can process the sample 384 within 8 to 10 hours of time. And important thing is compound treatment, cell lysis, digestion, everything happen in the Covaris plate. So plate to plate transfer is minimal, and it's also increase the sample recovery, because when you transfer from one plate to another, obviously the sample, we are going to lose the sample. And within 1 day, in our hands, in human hands, we can process 2 384 well plates. So that means we anticipate to increase the throughput up to 4 384 well plates by integration, the Hamilton, which we are acquiring within 2 months. So that means within 1 day you can process 4 384 well plates of samples. So we need that capacity to help the DMTA cycle. So then for the quality control, we look at what are these proteins from our new Astral Zoom mass spec for the deep proteome coverage. So we compare the Covaris versus no Covaris, and what you can see, it's a Venn diagram. What you can observe is we observe more peptides. This is the peptides. So for the proteins, we didn't observe that much of big difference, but the difference is coming from how many peptides we observe from the Covaris compared to the no Covaris. And as you can see, we observe more than 5% peptides. So we were curious, what are those 5% peptides? So actually these peptides are coming from the nucleus, which is actually benefit for us, because most of the interested drug targets we are caring about is the transcription factors, which is coming from nucleus. So it gives us much more added amount of peptides that we can look at to develop our methods. So it's added advantage for us. And this is the cellular localization bioinformatics, a bit of bioinformatics to check where these proteins comes from. So it's coming from mainly nucleus compared to the no Covaris system. Actually, it makes sense because Covaris is a sonication. So all the chromatin-bound protein, which are transcription factors, come to solution when you use sonication, because sonication helps to break down the DNA. So it's ideal if you are looking for nuclear proteins and transcription factors. And then we went to develop the targeted mass spec. So these are real samples. Put it to the mass spec and see the quality. As you can see, the analyte signal to internal standard. This is the targeted mass spec. So we know what we are looking for. So we want to know how much the target is there, how much target is degradation. So here just the amount of the target and replication, reproducibility, good reproducibility. And then this is the degradation curves, DC50, from these 384 well plates. And with this, we can, with the two 384 well plates, we can, within 1 day, we can profile 56 compounds. So the assay is now DMTA ready. And just to summarize, so as far as I know, this is the first time, someone has showed that you can do the digestion, everything in sample preparation in 384 in 1 day with this higher throughput, with the incorporation of 2-hour digestion. And because of that, the amount of reagents and the plate consumption is low. So it's so it give us the advantage of reduced cost and fewer samples to run by using next generation TMT. We are still exploring that at the moment, and compared to the 96 well plate, we have a higher throughput and automation ready, and also AI ready, and proteome coverage is we are reaching about the same as 96 well plate with sample consumption. And more importantly, nuclear protein, because we are caring more about the transcription. We have a lot of transcription factor programs, so we are very happy with the Covaris sonication methods to improve this target coverage. So now, we are ready with the DMTA assay, and we are impacting several drug discovery programs with this DMTA ready assay for several programs at this moment. With that, I would like to acknowledge Joanne, who has done almost all work. I'm just presenting, and also our group members, and also Covaris with the help and feedback given to us. Thank you.